Welcome to Biosumit. Today we'll learn about transduction. Now in my previous videos we have learned about the very basics of genetic recombination in bacteria, conjugation. Okay. Now transduction actually discovered by Zinder and Lederberg in 1951 in Salmonella. Now this phenomena happens through bacteriophage. Now bacteriophage actually acts as a vector to transfer DNA from one bacteria to another bacteria, or you can say donor bacteria to recipient bacteria. Okay, so before coming to the diagram, how it works, we have to know that transduction can be of two types, generalized and specialized. Here you can see generalized in the lytic is written under the generalized part. That means in case of generalized, the phase will undergo the lytic cycle, and for specialized, they will undergo the lysogenic cycle. We will see and show what are this lytic and lysogenic. Okay, now coming to the main diagram part, this is the diagram for the generalized transduction. Now see, there is a bacterial cell. Okay, and this is the bacteriophage, and this red one is bacterial DNA. I'm sorry, the viral DNA, the bacteriophage's DNA. Okay, now this is going to affect this bacteria. Okay, now what happens, the fast DNA and proteins will be made, and the bacterial chromosome, that is the blue one, will be broken into pieces. Okay, now in subsequently occasionally what happens, that during the phage assembly, pieces of the bacterial DNA are packaged, sorry, not the only bacterial, the viral one also, that will be packaged in a phage capsid. Here you can see this hexagonal part, these are the capsids. Okay, so you can see the bacterial DNA and the viral DNA both are packaged in the phage capsid. Okay, now if this part, see here is this part is containing the full bacterial chromosomal part, right? Now if this part is going to affect one new bacterial cell, so what will happen? So eventually this new bacterial cell will have the previous bacteria's chromosome, right? So that's how this DNA material, the DNA material of this bacteria is going to enter the new bacterial cell. Okay, and now in the normal process it will undergo its own reproduction, right? So it can be inserted into this, or it can lead to the same process like this. Okay, so that's how the horizontal gene transfer is happening from one bacteria to another bacteria. The genetic material is getting transferred. Okay, now this is generalized transduction. Now in case of specialized transduction, what happens, I have told you that it occurs during the lysogenic cycle of the phase. So what happens, here you can see this bacteria, this viral DNA is going to be inserted in the bacterial cell, right? Now in the next step what will happen, so in the next step you can see the viral DNA is here and the bacterial chromosome is also here. Then in the next step, this viral DNA is going to be inserted into the bacterial chromosome. Okay, like this. You can see the blue part. The blue part is the bacterial's own chromosome, and the red part is the viral DNA. So those two things are getting inserted. Now the same thing, it can create and packaged within the phage capsid. Okay, it can create like this coat. Okay, like this. Let's say this is the coat, and it is containing the bacterial chromosome with the viral DNA inserted. Okay, suppose this is, and eventually and occasionally it's going to be going to affect another bacterial cell and will lead to transfer its own chromosome or own genetic material to the new bacterial cell. Okay, so this is we can call as specialized transduction. Okay, so this is the main idea of the transduction, the general idea. Okay, So one main difference you can see here that in generalist transduction, some new phases, okay, like this, may have no viral DNA. You can see here there is no viral DNA. Okay, but in case of specialized, it must have the viral and bacterial DNA both, right? Or in the most cases you can say if it breaks in the middle, that can be the different scenario. Okay, so this is the general idea of transduction and its parts. Hope you have understood the part. Thank you.